everyone. Good morning to everybody. And the topic that we have taken today is the fifth chapter in your grade 12 called Genetics. And the name of the chapter is Principles of Inheritance and Variation. So before saying about the principles of inheritance and variation, what is inheritance? I have inherited my father's property. That means what? Whatever was there with my father, I got some property. So inheritance. So we have the maternal body and we have the father's body. That is called, as when you take about the paternal and the maternal, both combined together is your genes. So your gene is a recombination. So whatever, you look like your father, you look like your mother, nose looks like your father, ear looks like your mother. A lot of different terms we say. Okay, the speech is like your father, you like your grandfather, you like your great grandfather in your dressing. So all this comes into a major factor. So what is this controlling factor? Means today we call them as genes, and more in olden time they call as factors. So when you look about genetics, it deals with the transmission of character from one generation to another generation. To summarize this, our chapter is classified into two different branches. The first branch is called as classical genetics. The first one is the classical genetics and next one is the modern genetics. Classical genetics and modern genetics. So the first thing is classical genetics. In classical genetics, we are going to discuss about a small cross, or in other way, here we have an important person called as Gregor Johann Mendel. He was working with an experimental plant. The experimental plant is Pisum sativum. And you have to underline the genus and the species. And the common name for Pisum sativum is garden pea plant. And why you have taken it all will be dealt in the next session. And about this, we are going to discuss about some important crosses here. What are the important cross that we are going to discuss in this chapter means? The first one is Monohybrid Cross. Monohybrid Cross. Based on this Monohybrid Cross, two laws were framed by Gregor Johann Mendel. What is the first law is law of dominance. Law of dominance. About that it will be discussed. The second one is called as law of segregation. And there is another name for law of segregation, that is called law of purity of gametes. Law of purity of gametes. Then he performed another cross called as dihybrid cross. Based on that, he formulated a law called law of independent assortment. Law of independent assortment. And there are some important crosses also that we are going to discuss here. First one is called as test cross and next one is called as back cross. So there are test cross and back cross, some interesting cases that we are going to see. And apart from it, there are some interesting cases also. What are the interesting cases? The first interesting case is incomplete dominance. Law of dominance, here something called as incomplete dominance. About that we are going to discuss. And the next important thing we take into consideration, it is called as pleiotropism. The next one is called as polygenic inheritance. Okay, so it's over now. No, my dear, it's not over. After polygenic inheritance, yeah, and about polygenic inheritance. So these are some important 3-mark questions which is there for our board exams. Incomplete dominance, the help of Snapdragon, you're going to prove it. And pleiotropism, polygenic inheritance. Apart from it, you have other rules also. What are they? They are multiple allelism, and the next important thing is codominance. So these and all we are going to learn in classical genetics. So what we are going to learn in modern genetics? In modern genetics, here the father of genetics is Gregor John Mendel. Father of modern genetics is nothing but, it is called T.H. Morgan, Thomas Hunt Morgan. He was working with an simple fly called as fruit fly called as Drosophila melanogaster. Commonly we call them as fruit fly. Why I have chosen that, we are going to see. Here he performed a cross, only a dihybrid cross based on something called as a linkage and a recombination. Linkage and recombination. Okay, fine. What are the other things that we're going to discuss in this? So what we're going to discuss in other parts here is, here the next important topic that comes into our mind is sex determination. Okay, what are the different types of sex determination that we're going to take into consideration? XX, XY type of sex determination. Number 2, XX, X0 type of sex determination. ZZ, ZW type of sex determination. And next is diplontic or haplodiplontic sex determination. 4 types of sex determination we are going to discuss. Apart from it, we are going to discuss about Mendelian disorder. Mendelian disorder. After discussing about Mendelian disorder, we are going to discuss about genetic disorders. Very interesting topics, and you will love to learn these topics. And apart from it, and concept is there for you with the pedigree analysis. So these are the topics that lies for us in case of modern genetics. In this, very important things about why I have chosen Drosophila melanogaster, linkage and recombination. The important things are, these two are important sex determination methods which is mostly asked for 2 marks in your board examination. And look about Mendelian disorders. All are very important. We cannot skip any Mendelian disorder. We cannot skip genetic disorder. Pedigree analysis is rarely asked for 1 marks only. So now nothing to discuss more about that. But very important, where we have to give more importance is, as per the survey of past 5 years, we have to give more importance to classical genetics and topics which are highlighted. All these areas are very important for your board examination. Now in the next chapter, next topic, we'll be seeing about the basics and Mendelian approach. Okay, so with this we're going to stop this class, and next class we'll meet you children. Thank you very much.